my dear little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today it's wrap up time. We are going to have a rapid revision of one short video on sexual reproduction in flowering glands, where as I said, whatever we covered in a week, everything is going to be covered here as well in a very fast pace. So while I was preparing the notes, I could remember myself being as a student. When I was a student, in every gang there used to be one friend who saves our life before exam, right? We just go sit, listen to them, and then whatever they say, it's a sort of rapid revision that they give, and whatever they say, it just stays in our mind, and then we go write in our paper. So this is going to be exactly like that, where I will be I'll be telling lot of things, lot of information. I keep rest assured that you have already prepared all this thing, and this is just for your revision purpose. Now, if you're not just revising, you want to study it, then you can try, but I recommend watch the longer videos. That will be much better, which we have already made. It gives you more in-depth information, but still you can try this out as well. Now, starting with the structure of the stamen or the microsporangia. So children, you know stamen, it is the male reproductive part of the flower. Okay, now over here, this particular structure that you see, the lobed structure, what do you call it, children? You call it anther. Next thing that you see over here is basically a stalk-like structure. What do you call it? You call it filament. And now the anther and the filament, basically it is attached to the lower part of the flower, which is known as thalamus. Sometimes this particular thing attaches on the thalamus. Sometimes this particular filament can attach on a petal as well. Okay, there are two conditions. Sometimes it attaches over the petals, most of the times it is there on the thalamus. Now this area of the filament which is closer to the flower, you call it proximal region, and the one that is distal, in distance to the flower, you call it what? Distal region. This has come. What is there in the proximal region of the filament? It is the thalamus. What is there in the distal region of the filament? It is the anther. Now if you talk about anther, you can see it clearly. It has two lobes, 1 and 2, which is why I call it bilobed. And I also call it dithecus. Why do I call it dithecus? Because if I take this anther, I cut like this and I see from above. What is a view? I look at something like this. Over here, this is one lobe, this is second lobe, and each lobe has two thecas. This is one theca, this is two theca, second theca. Similarly, over here we have one theca, we have another theca. So in total we have 4 theca. In one side you have 2, which is why you call it dithecus. What do you call it, children? Dithecus. Okay, so anther is bilobed and dithecus. Going ahead, what is the structure of the anther? What is there inside the anther? So this particular structure that you see in green, what do you call it? You call it the microsporangium. This is where the pollen grains are going to be made. This is where the pollen grains are going to be made. Okay. Now, outside the microsporangium, you have different layers. Let's start with the first layer. The outer layer, the white one that you see over here, it is known as the epidermis. Going inside that, the blue one that you see over here is endothecium. Going inside that, we have middle layer over here. Now, middle layer is in between the epidermis, endothecium, and one more layer which is going to come, which is known as tapetum. So, if you see, the microsporangium in total is covered by something that is known as tapetum. That is the first layer that covers the microsporangium. What is the use of tapetum? It provides nourishment. It provides nourishment to the sporangium, okay, microsporangium, and it is also, I'll tell one more feature about it. If you see its cell, it has 2 nuclei. If you see its cell, it has 2 nuclei, which is why I call it binucleate. So basically, its nucleus divided, but it didn't divide into 2 cells. So still, they have 2 nuclei in one particular cell, which is why I call it binucleate. Okay. So epidermis, endothecium, and middle layer provides protection, whereas when you talk about tapetum, it is responsible for nourishment. Now, one more thing about epidermis, endothecium, and middle layer is that they are going to help in the dehiscence of anther. What do you mean by dehiscence of anther, children? Anther, it dehisces, it opens up, and that's where the pollen grains which are made up here slowly comes out. Okay, slowly it comes out, which is what is known as dehiscence. Okay, fine, done. Now, can we talk about just this one as a whole? Let's go ahead. And over here I have written also what is there over here. Even though I call it microsporangium, this microsporangium structure, it is made up of several things. The inner cells that you see over here in total green color, they are known as sporogenous tissue. What do you call them, children? Sporogenous tissue. Now we go ahead and talk about sporogenous tissue. Over here, all the cells that you see over here, they're all smiling. Okay, all of them are known as pollen mother cell, or you call it megaspore mother cell. When do you call someone mother? when they are going to give birth, right? So over here also they are going to give birth to pollen, or I can say that they are going to give birth to megaspore. So megasporangium, okay, it has sporogenous tissue in the inside, and each cell of the sporogenous tissue has the capacity to become a mother. Is the mother, okay, is the mother. It's the pollen mother cell, or you call it megaspore mother cell. Now I take one PMC over here, one smiling PMC over here, and then I tell it to undergo meiosis. Okay, I tell it to undergo meiosis. Why meiosis, children? Because all the cells over here, they are diploid in nature. What are they? They are diploid in nature, but gametes, they have to be haploid. If you talk about gametes, they have to be haploid. Why they have to be haploid? Why should they contain half the number of chromosomes? Because you only think, again I'll tell you with the example of human beings, 23 if we get from both our parents and father and mother, okay, then basically the child will be having 46 number of chromosomes, and this is the normal number of chromosomes that we have in our cell. But if the father's gamete didn't divide, it was 46, from mother's ends also you are getting 46, together it's 92. This is not the normal cell chromosome, number of chromosome, which is why if we half it and then send it, probably things would be better. So same thing happens over here. The gametes which are going to be formed should be having haploid number of chromosome, should be having, children, haploid number of chromosome. So they are diploid. To make them haploid, they have to undergo some process which is known as meiosis, or it is also known as reductional division. What do you call it? Reductional division, because here the number of chromosomes are going to reduce, children. Okay, so meiosis basically occurs in two stages. There is one stage, okay, then there is second stage. Both them together you call it one total cycle of meiosis. So one total cycle of meiosis has two stages. So in one stage the PMC underwent division, now you have two cells. Second stage, second meiosis happens, basically you have 4 cells. Now I call these 4 cells as 4 microspores. I call this 4 cells as what? 4 microspore. This was, sorry, did I write megaspore anywhere? Yes, sorry, I meant microspore. Okay, so basically the microspore mother cell, that is PMC or pollen mother cell, is there. Okay, it underwent division which is known as microsporogenesis, and now at the end you have so many microspores. How many, children? 4 of them. 4 microspores are there. Now 4 microspores, if I want to call them together as a family, I call them microspore tetrad. Okay, microspore tetrad means 4 microspore. Now this microspores, they feel that they are in a new world, so they have to be protected. So they are protected with the help of another thing which is known as callose. Please remember it, it has come. Okay, it is known as callose. Now there is an enzyme that is known as callase which breaks this callose down later on. That's a later process, we don't need to go deep into that. But just understand these 4 microspores which are there are covered by something that is known as callose. Now at the end, what will happen to this microspore? What is this microspore actually? Well, these microspores are nothing but the pollen grains. These microspores are nothing but the pollen grains. They are the pollen grains. Okay. So basically, who form the pollen grains? You have sporogenous tissue. The cells of the sporogenous tissue, each of them is known as PMC or MMC. They undergo meiosis, which is reductional division. At the end, you get 4 microspores. Together, you call them microspore tetrad, and this whole process is known as microsporogenesis. And since they are all together clumped up and they have a wall around them, or basically a layer around them, you call it, it is formed of callose. Okay, you say it is formed of callose, and now these cells which are going to arise from each microspore are known as pollen grains. So how many pollen grains will you get from one PMC, children? You get 4 pollen grains from one PMC. Now let's see the structure of pollen grain. Over here, the thing that you see over here is a pollen grain. Okay, now let's see. There are inside the pollen grain, inside the pollen grain, there was a cell. This PMC underwent an division, and now it's a microspore. Agree? This microspore divided once, okay, inside, and now there are 2 cells. One cell is over here and another cell is lying over here inside the cytoplasm of another cell itself. So the one that is lying is known as generative cell, okay, and the one inside which this particular cell is lying, it is known as vegetative cell. This is because of first mitosis. First mitosis happened. Okay, so after meiosis, after it is formed, now when I say it's a pollen grain basically has 2 cells. Okay, pollen grain basically has two cells, and these are the cells that I am talking about. One is vegetative and the other one is what, children? Generative cell. Okay, this happened because of mitosis 1. Mitosis 1. Please remember it, mitosis 1. Okay, and now if you see the layer, total layer, so this inner white layer, I don't know if I have mentioned it. Yes, this particular white layer that you see, it is just the plasma membrane of the cell. It is just the plasma membrane of the cell. Now outside it you see a blue color thing, right? You see a blue color thing which is known as intine, and intine is made up of pectocellulose. It is both made up of pectin and cellulose, which is why I say pectocellulose. Now outside that you see something that is known as exine, which is green in color. Exine is the that particular layer which can withstand anything. Why can it withstand anything? Because it is made up of something that is known as sporopollenin. It is a very tough chemical that is out there. It can endure through acids, alkali attack, anything. Okay, so you have your intine, you have your exine. Exine is made up of sporopollenin, intine is made up of pectocellulose. Inside that you will be having a generative cell and also a vegetative cell. This is a result of mitosis 1. This thing that you see over here is a result of mitosis 1, and this pores that you see in between the exine layer, what do you call them? Germ pores. With the help of germ pores only, later on the pollen tube will be formed. Where is the germ pore? Okay, from here only the pollen tube will be formed and the male gamete which is there will be coming out. But you might be thinking, where is the male gamete? We didn't study about male gamete itself. I'll tell you. So 60% of the angiosperms, that is the flowering plants, they go in this stage only. The pollens are shed in this stage only, where they have generative cell and vegetative cell. Okay. Now once this pollen goes and lands on the stigma, it slowly undergoes the second mitotic division. This was mitosis 1, right? The second mitotic division happens over here. I'm writing. Mitosis 2, it happens over here, and then as a result what do you get? You get 2 male gametes, children. What do you get? You get 2 male gametes. So 60% of the angiosperm, they go like, they do not divide. The generative cell is as it like that, vegetative cell is also like that only. They go there, land there, and then later divide into what, say 2 gametes. So in this stage, if they are not dividing, can I say that it is going in 2-cell stage? Yes, one will be generative and the other one will be vegetative cell. Now 40% of the angiosperm, rest 40% of the angiosperm, they go in 3-cell stage. Ma'am, how 3-cell stage? Basically, they do not wait for going and landing there and dividing. They just undergo the division over here itself. So you have 1 cell, you have 2 cell, you have 3 cell, which is why I have written 3-cell stage, where one is the generative cell which has divided into 2 male gametes and the other one is the vegetative cell. Okay, so generative divided into 2 male gametes and one is the vegetative cell. 2 plus 1, 3. You have what? 3. Okay. Now, children, the pollen, the mighty pollen grain says, I am the male gametophyte. Why does it say I am the male gametophyte? So, gametophyte means that particular cell which can give rise to gametes. So, over here, don't you think that this is a pollen grain and it is going to give rise to gametes, that is this? Yes, which is why pollen grain is the male gametophyte, children. Please remember, pollen grain is the male gametophyte, and as I said over here, 2 mitosis is there, one for dividing into generative and vegetative and the other one for dividing into 2 male gametes. Okay, will you be able to remember it? If you are not able to remember it, please write it somewhere. Fine. Okay, going ahead, pollen energy storage and viability. Let's see. Pollen energy, lots of people have pollen energy when they have pollen around the wind. They like things like that happens. Okay, some people they have asthma also, some people they have a bronchitis also because of the same thing. Now the main villain which is given in your NCERT textbook said that there is something that is known as parthenium or carrot grass. Okay, there is something that is known as parthenium or carrot grass which causes a huge lot of trouble. It is a huge cause of pollen allergy. Okay, now let's talk about pollen tablets. People have pollen tablets also as food supplements, as what, children? Food supplements, and it is taken by athletes for increasing their vigor. Okay, for increasing the energy source. Fine. Going ahead, pollen viability. What do you mean by viability of pollen? Basically, a pollen is there, it has to go land on the stigma and then from the germinate and give rise to pollen tube also. This is known as viability. Okay, so I have written ability to germinate and form a pollen tube. So basically, there are certain species which do not have much viability. Okay, the pollen grains are going, but within 30 minutes, if it's not landing on the stigma, if it's not all going good, then probably they are of no use. So, example is rice and wheat. they just have 30 minutes of viability. Just 30 minutes of viability. After that, rice and wheat pollen grains will say, I cannot go and do pollination. I cannot let pollen tube formation happen there. Okay. But on the other hand, if we talk about Rosaceae, Leguminosae and Solanaceae, the families, okay, basically they are viable for months. They are viable for months. What is it, children? Right, left side, I remember like that. Okay, you can remember in your own way. Rosaceae, Leguminosae and Solanaceae, they are all viable for months. Whereas talking about rice and wheat, which we normally have, just think that rice and wheat, after we have it and then after 30 minutes we start feeling hungry again. It's like that. Okay, it's like that. I know it might not be the case for you, but it's the same for me. Okay, so that is the thing. Yeah, going ahead, if you want to store pollens now, okay, you're worried what will happen to this particular species if it dies now. I should store the pollen. So we can store the pollen, and as you can see over here, the pollen is kind of petrified because it is saying that it is minus 196 degrees Celsius up here. It's too cold, I cannot stay here. And this is what, children? This is liquid nitrogen. In liquid nitrogen, pollen is allowed to store. Okay, minus 196 degrees Celsius is the temperature. Now this can be used for crop breeding program later on, such that we do not have any sort of extinction and the plants are still there. Works? So this is all about your megasporangium. I hope it is understood. Megasporangium has sporogenous tissue. Sporogenous tissue, each cells are PMCO or micro. Did I say megasporogenesis? Oh my god, sorry. Microsporogenesis. Okay, please understand. Micro. Okay, think like that. Okay, so basically microsporogenesis has microsporangium has sporogenous tissue. Each sporogenous tissue has microspore mother cell or pollen mother cell. Each of them divides and gives rise to 4 cells with the help of meiosis. Each 4 cells that you see over here will undergo mitosis to form the pollen grain. Pollen grain has 2 cells, which is known as generative and vegetative cell. Generative cell will again divide further into second mitosis to give rise to 2 male gametes. But 60% of angiosperms, it goes in 2 cell stage only. 40% of angiosperm goes in 3 cell stage, but the division has already taken place. The outer layer of pollen is known as plasma membrane. Outside that, intine, which is made up of pectocellulose. Outside that, exine, which is made up of what, children? Tell me in the comment section or anywhere, chat box, anywhere. It is known as sporopollenin. Okay, it is known as sporopollenin. It's a very important chemical that you need to remember. Whatever comes, rain, sun, alkali, acid attack, anything, it can still survive. Okay, and then there are pores in between the exine, which is known as germ pores, through which the pollen tube comes out. And this is the thing that we have to study. Please remember this is example. And minus 196 degrees Celsius. Done? Talking about the female reproductive system, let's start. So over here we have something which is known as pistil or carpel. You call it pistil or you call it carpel. As you know, it is having a structure which is known as stigma. It's a landing surface where the pollen grains come and land. Next thing, we have style, a long tube, and then there is ovary. In this enlarged structure that you see over here, it's the ovary. Inside the ovary, you have something which is known as ovule. Okay, inside the ovary you have something that is known as ovule. Now let's get inside the details of everything. So over here, if you see something, this is known as placenta. Okay, and this placenta is going to provide nourishment to the ovule with the help of attachment which is known as funicle. Which is known as what? Funicle. Now the site where the funicle is attached, I have to zoom in, and I don't think I need pen over here, so I'll show you. You can see it clearly. Okay. So this is placenta, and this placenta is kind of attached to what? The ovule. Now there is a scar that is being formed because of this, and this is known as hilum, children. It is known as hilum. Okay, this scar remains whenever you see a seed, you see a small pore sort of thing, that is the hilum. The scar that is being formed, that is the hilum. Now going ahead, if you talk about the ovule, this whole thing, the blue thing that you see over here, it's the ovule. And as you can see, there are 2 layers protecting the ovule. One is the inner one is the inner integument. The other one, the outer one, is known as outer integument. But if you see clearly, the inner and outer integument are absent in this particular region. What do you call this particular region as? You call this micropyle region or micropylar region. This is the point from there where the pollen tube will come and get inside the ovule, because ovule will be having the female gamete. The female gamete has not yet formed. We are going to tell how it is being formed over here. Okay, so I call the ovule as megasporangium. What do I call it? Megasporangium, because it is going to form the this string, that is the female gamete later on. Okay, similarly in anther also we said the anther has the microsporangium. Similarly, as of here, I can say the ovary has the megasporangium, and this megasporangium is nothing but your ovule. Okay, now if you see this there, okay, so you spoke about micropellar region, there is an opening, right? Now on the other hand, the opposite side of the micropellar region, I can call it the chalazal region. Okay, so if this is the ovule, this side, if it is micropellar, then this side it has to be what, children? It will be chalazal region, or it's a chalazal end, always opposite to the micropellar region. Works? And the cells that you see over here, there are so many cells over here. One I have colored it differently, okay, but the cells in total, it forms something that is known as nucellus. So one cell in this tissue, just like we had sporogenous tissue, there were so many cells, and all of them were PMCs or MMCs, right? But over here, in the case of nucellus, only one guy is megaspore mother cell. Only one guy is megaspore mother cell, and we are going to talk about megaspore mother cell later on. Okay? So children, please understand, in the case of females, the ovule is known as microsporangium. Fine? This end through which pollen tube comes in, it is known as micropellar region. The opposite one, it is known as what? Chalazal region. Okay. Now, ovules, there can be one. Okay, there are flowers where the ovules are just one. Example, paddy, mango, wheat, etc. But there could be areas where the ovules are more in number. For example, when you eat watermelon, you see so many seeds, right? So it has multiple ovules. If you talk about papaya, it also has multiple ovules, right? Now there is orchid, which probably you have not eaten because it's a flower, but it also has multiple ovules. Okay, done? Is this particular thing clear? Going ahead. Are you? Why is it there? Just a second. Little bit of confusion is here. Okay. Now, listen, pistil can be so many in number. Pistil can be one, pistil can be so many in number. When there are so many pistil, we call it multicarpillary condition. Where is it? You call this condition as, children, multicarpillary condition. So over here you see multiple carpels, over here also you see multiple carpels, even though if you are not able to identify, I am telling you, they have multiple carpels. Okay. Now this carpels, when they are all fused together, you call this as syncarpus. You call this as syncarpus, and you can see it papaver. You remember papaver? We studied in human health and diseases. Papaver somniferum, if you remember. Okay, opioids are being found by this. Now here, as I said, the carpels, they are fused, children. They are multicarpillary condition and they are fused. It is known as syncarpus. On the other hand, over here you see this is one carpel, this is second. So all of them, they are not fused. They are separate. They are free. You call this condition as apocarpus. Where should I write? Yeah. You call this condition as apocarpus, if you remember. And what is the example? Your best friend, Michelia. Can remember like that. Michelia is your best friend. That is apocarpus. Separate thing. Okay. Now, there is a type of ovule that is given over here. You call it anatropous ovule. Anatropous ovule. And over here, there is a slight change. So, in the picture that we just studied, chalazal end was here. And here only you had the hilum, the funicle and everything, right? But this is a slight change. The ovule has turned like this. Okay. Now, there will be placenta somewhere over here, and there will be funicle somewhere over here, as you can see, and then there will be hilum here in the micropellar region. So your hilum has nothing to do with the chalazal pole. Just understand that. Where is it? Yeah, your hilum does not have anything to do with the chalazal end. Okay, for example, if the placenta is somewhere here, the funicle will be like this. So that is known as anatropous ovule. Anatropous ovule. And what was the example, children? Over here, the example was also given. They have not mentioned the example. I don't know. Okay. Then, yeah. So then it was not asked. Okay. I'm sorry. Anatropous ovule is there. Okay. I hope that is also clear to you. Now we are going to talk about this guy that is over here. Okay. Our MMC. Let's see. So this process is known as megasporogenesis, because obviously the megaspores are going to be formed. I am the one who will form megaspore, the white one is saying. Okay, I am the megaspore mother cell. It will again, as I said, the same thing that happens there. Okay, just understand that the cells of the sporogenous tissues were used. Here the cells of the nucleus is used. Anyways, this is a diploid cell. This is a diploid cell, that is 2n. Okay, there also it is diploid, that is 2n. Now it has to undergo division and make n. Now let's see. So it has divided, meiosis 1 has taken place, reductional division. Now you have 2 cells. Again it has divided into 4 cells. But I see something. 3 of them are sad and 1 is happy. Why? Because they degenerate. All of them that you see over here, they degenerate. Only one sustains, and you call it the functional megaspore. What do you call it? The functional megaspore. Since we had 4 megaspore over there, the 4 were like this. The boys gang was really strong. The girls gang went really bad. Over here, only one friend is there. Rest all of them, they got away. Okay, they degenerated. So there is only one functional megaspore that we have over here. And let's see what happens to this functional megaspore. So this functional megaspore, I'm going to make like this. This whole structure is the ovule. Okay, this is the ovule, children. Inside that there is a cell. There is a cell with a nucleus. Let's see what happens to the cell with the nucleus. So there also, please compare and learn. There also microspore was there, and this microspore was forming what? The pollen grain. In that it underwent 2 mitotic division. But over here we girls are going to make them loose, because we will undergo 3 mitotic divisions. We will undergo 3 mitotic divisions. So let's see what happens. First, understand that functional megaspore, there it was forming the pollen grain. Here it is going to form the embryo sac. If pollen grain there was the male gametophyte, embryo sac here is the female gametophyte, because this is where the female gametes are going to form. So over here it has a nucleus. This nucleus, cell does not divide initially, the nucleus divides. Over here you have 1 division, 1 mitosis. Then second division, second mitosis. Then third division, third mitosis. You have 8 nuclei at the end. At the end you would have 8 nuclei, and then cell wall will be forming around it. Okay, cell wall will be forming around it. 3 of them arrange here, 3 of them arrange here, 2 nuclei just arrange here in between. 2 nuclei just arrange there in between, rest 3 and 3 arrange over here. So total you have what, children? 8 nuclei. You have 8 nuclei. But what is the number of cells that you have over here? Basically 1, 2, 3, 1, 2, 3, 6. And then this whole thing that you see over here, which I have colored in blue, this is known as central cell. So I can call it that it is in 7-celled stage, but there are 8 nuclei. But there are 8 nuclei. Okay, now let's see the name. So over here, this one that you see, the orange one, it is the female gamete. So we have got the female gamete, children. Now beside it, we have 2 of their best friends, synergids, who are going to die for this particular friend of them, that is the female gamete. So there is synergid cell, and synergid cell has like something like this, which is known as filiform apparatus. The use of it is to, basically when the pollen tube comes, they say come here, go through us. So the pollen tube comes and basically goes to the synergid. The synergid would die later on. I will tell the story, it's a sad story. But as of now, just understand you have 2 synergids and they have filiform apparatus. And in between, what do you have, children? You have central cell, and the central cell basically has 2 polar nuclei. These nuclei are known as 2 polar nuclei. Okay. Now over here you have 3 antipodal cells as well. This area will be the micropylar region. This area is the micropylar region. So you will see the antipodal cell in the chalazial end, and this in the micropylar region. Done. Also one more thing, the embryo sac is known as the female gametophyte, because here the gametes are formed. Also understand there was just 2. Okay, one more thing. Here, as you see, only 1 megaspore went ahead. So which is why I call it monosporic development. Okay. Now over here, this monosporic development will lead to these many cells. Okay, and this is the female gametophyte and monosporic development, and then there will be successive 3 mitotic division. Okay, this is an important MCQ. I have made you do that, but still. Now let's talk about pollen pistil interaction. What will happen? So basically, there's a pollen that came. Okay, pollen had the right chemicals on its surface. Similarly, stigma also has receptors. Now on these receptors, the pollen has to land and see that everything is if it goes well, the pollen will germinate and then it will form the pollen tube. But on the other hand, if the receptor and them are not matching, then this pollen germination and pollen tube formation will not happen. So usually this condition happens when Intraspecific pollen comes in. Intraspecific pollen as in pollen from other species comes in. Second thing, when there is self-incompatibility. Incompatibility. When there is self-incompatibility as in their own anther producing pollen grains and it landing on the own stigma. Why it becomes incompatible? This is genetic mechanism. Plant does not want this thing. Plant does not want self-pollination to happen, because this will lead to what? Inbreeding depression. This will lead to, plants also have depression, which is known as inbreeding depression. No children, it's basically inbreeding depression is when the fusion happened, everything happens, still there won't be fertile seed being produced over there. This is known as inbreeding depression. So we do not want that. To avoid that, let's do self-incompatibility. We are not compatible with each other, and I'm pretty sure of that because we are from the same family. So it's not possible. Okay. But intraspecific is there. If some other plant wants to donate its pollen, that is also possible. But usually plants just show this thing that is known as self-incompatibility. Now, suppose the right pollen lands on the stigma. What will happen? So 60% of angiosperm which are coming with generative cell and vegetative cell, they come, land on it, form the pollen tube, and now the generative cell divides into the two male gametes. Okay, the other one, they are coming in the 3-cell stage only. But ma'am, where is the vegetative cell? Bachcha log, vegetative cell is the one that is going to form the pollen tube. Very important MCQ. Vegetative cell, which you see over here, as I've written, vegetative cell forms the pollen tube. It is the one that will form the pollen tube. And now this growth is known as apical growth because it's happening at the apex and it's still growing inside, and it is also known as chemotropic movement. Why do you call it chemotropic movement? Because this pollen tube is like not where to go, but there is someone sitting over here and releasing chemicals, so it is going in that particular direction. So chemotropism, positive chemotropism is being shown over here. Now for doing this particular action, they need the help of two ions which are known as boron and calcium. One is boron and the other one is calcium, which helps in the pollen tube formation. I was saying test tube. Okay, now what will happen? I'll tell you the story. The male gametes came and it came to the synergid. Okay, it came to the synergid. Now what will happen to the synergid, children? Once it comes, I hope I have a diagram. Yeah, so over here it came. Before it enters the synergid state, degenerate. Before it enters, the synergids that you see over here, they get degenerated. It's a very important question again. So the filiform apparatus, it comes to synergid. Synergids gets degenerated and it has the two male gametes. One male gamete go fuse over here with the female gamete, and the second male gamete fuses with the two polar nuclei. It says, what will I do? I also want to fuse. I'll go fuse with the two polar nuclei at the central cell. Okay, so first condition is where the male gamete and the female gamete is fusing, right? And this will lead to formation of what? Children, zygote. It will form zygote. Now male gamete, female gamete both are N and N. Zygote will be what, children? 2N. You got diploid zygote, right? This process is known as syngamy, or it is known as fertilization. You call it syngamy or you call it fertilization. Second thing, the male gamete that is there, it goes and fuses with the two polar nuclei over here. Now, how many nuclei do they have? Male gamete has a nucleus, there is two polar nucleus. So, we have 3 nucleus altogether. Since there is a fusion of 3 nucleus, you call this as triple fusion. What do you call this as? Triple fusion. And what are they going to make? We made the zygote. Well, to nourish the zygote, you need something that is known as endosperm. So, the ones that will give rise to endosperm are found with the help of this. So, basically what is happening? You have everything, you have the male gamete and the two polar nuclei. It is going to give rise to primary endosperm cell, which has the nucleus which is known as primary endosperm nucleus. Okay, it has the primary endosperm nucleus. All good? All good? And now the primary endosperm cell later forms the endosperm which will provide nourishment to the zygote. And this is known as triple fusion. So now you see 1 and 2. Twice the fusion is happening, which is why you call this as double fertilization, children. What do you call it? You call it double fertilization. I hope this is also clear. We studied this, right? So please remember this, PEN is formed, that is a primary endosperm nucleus is formed, and this is the part of what? Primary endosperm cell. Now later image do you have? Yes. Now you have zygote, children, and you have primary endosperm cell. Now endosperm loves zygote so much. Motherly care is there. Endosperm will say that zygote, I want to see you to develop into an embryo. For that, I will give you everything. I will provide nourishment. I will provide nourishment. And it forms before the zygote develops. Before the zygote starts developing the embryo, developing into the embryo, the endosperm forms. This is a very important MCQ again. So what happens to the endosperm? How will it form? This is just one cell which has 3 nucleus. Okay, so I say this is what? Triploid. This endosperm that you say, it's a triploid tissue because it has 3 nucleus. N plus N plus N gives you what? 3N. Fine. Now, primary endosperm nucleus cell, okay, inside that it has nucleus. This nucleus starts dividing as usual every time division happens, right? So there will be free nuclei. This is a stage of endosperm formation. You call it free nuclear endosperm. Later what will happen? These nuclei which you see, it will be surrounded by cells. And now it will get cellularized. It will form cellular endosperm. Now you can see free nuclear endosperm in the case of coconut. Okay, in the coconut, the liquid thing that you see over here, it's the free nuclear endosperm. Okay, it is not bound by any cell membrane, nothing, cell wall, nothing like that. But now if it is bound by cell membrane, it forms the cellular endosperm, which is the white thing over here. The kernel that you eat, the white kernel that you eat is basically the cellular endosperm, which will later help in the formation of what? Basically slowly, why is nothing not working? Yeah, slowly the embryo will also form inside. People also eat embryo. Okay, so please understand that there is a primary endosperm cell. It has a nucleus, primary endosperm nucleus, which is triploid in nature. This nucleus undergoes division 1 to 2, 2 to 4, 4 to 8, things like that happens. Okay. Now, this nuclear, free nuclei, they are all good, which you call as free nuclear endosperm. Later on, cell wall and everything surrounds it. Same typical life that they have. Cellular endosperm is formed. In coconut, you can see free nuclear endosperm and also cellular endosperm. Okay. Now, this is for nourishment of the zygote, which is why it forms way before the zygote develops into an embryo. Now, it should not come. It has come. So, I'm going back and I'll arrange in this and give you. Okay. So, now, there is embryo. Zygote is also going to form embryo. There is zygote, single cell zygote. It will now divide into 2. Now, you have to remember the flow of this. So, there is zygote first, children. Then it forms something that is known as pro-embryo. Then it forms something like this, which is known as globular embryo. Then it forms heart-shaped embryo. And then it forms mature embryo. So, don't feel dizzy after seeing these images. I'm going to make your life really easy. Zygote, pro-embryo, globular. It has a globe-shaped thing, right? And it is heart-shaped, opposite heart, ulta heart, okay? Heart-shaped embryo, and then there is mature embryo. So, usually, this is common for all different type of plants. The angiosperms, I'm talking about them, okay? The flowering plants. This is common, but towards the end, it could be different as well. Now, let's talk about, why did this come? What did I click? Okay, sorry. Yes, now let's talk about the seeds, which are seeds, as in I'm going to talk about the embryo which is there also in the seed. Okay, so there are 2 types of seed, children. One is dicotyledon seed and the other one is what? Monocotyledon seed. Okay, inside seed, what do we have? Inside seed, what do we have, children? That is a major thing. I skipped that part. Okay, listen to me very carefully. So, once fertilization is done, ovary forms the fruit. Okay, ovule forms the seed. Inside the seed, what do you have? You have the embryo, okay, and endosperm sometimes, okay. So, now we are going to talk about this particular embryo, okay. And why did I say sometimes endosperm is there, sometimes it's not there? We are going to see that out. So, there is dicotyledonous seed and there is monocotyledonous seed. Example of dicots, pea, gram, chana, everything that you can break into 2 are known as dicots. Okay. On the other hand, there is monocotyledonous seed like wheat, maize, rice, barley, castor, coconut, everything that you cannot break into 2. Do not say that I can break coconut into 2. No, I'm not talking about that. Basically, there is a thing that you break into 2. You break into coconut into 2 with the help of knife, right? It naturally does not get broken like that. So, that is something different. Now, listen to me. Over here, this is a pea. Okay, over here, this is a pea that is there. It says we use full endosperm. So, endosperm which was there for my growth, my zygote's growth into embryo, I use it fully. No endosperm is left. But when I do it, I am going to store this endosperm in my embryo in a form of something else, which is known as, children, cotyledon. What do you call it? Cotyledon. Do you want a zoom of this? I can give you a zoom up image of this. Okay, this is known as cotyledon. So basically what has happened, the endosperm which was there for its growth and all, it took all of a sudden everything and it stored in itself only. This embryo is storing in itself only. This is now known as a cotyledon. Okay, so dicots are like that. So the pea and everything, they are like that. But on the other hand, when you talk about monocots like wheat, maize, rice, barley and things like that, they say we have little endosperm left. We do not want it to consume it completely. We don't need it as of now. So we have little endosperm still left. Now the seeds where the little endosperm is still left, you call them albuminous seeds or you call it endospermic seeds. Okay, you call them albuminous seeds or you call them endospermic seeds. Where you do not have endosperm left at all, you call them non-albuminous or you call them non-endospermic, not spermis. Okay, you call this non-albuminous or basically non-endospermic. Okay, wait. Now we are going to talk about what do they do with the this thing, nutrition and all which they got from the endosperm. So this is an example of a dicot. Okay, I have not mentioned here. This is a dicot embryo and this is a monocot embryo. So inside the dicot seed you will find a dicot embryo, inside the monocot seed you will find a monocot embryo, right? So over here they have an axis like this, towards two sides they stored everything like this. Now they have two cotyledons. Okay, now inside that they will be having an embryonal axis like this. The upper part which is above the layer of cotyledons, you call it epicotyl. You call it epicotyl, and this lower area which is below the cotyledons, you call it hypocotyl. Now at the end of the epicotyl you find something which is known as plumule, and this plumule is the future shoot. It is going to form the shoot. On the other hand, at the tip over here, okay, tip of the hypocotyl, you find radicle. What is radicle, children? It is a future root. And this radicle which is there over here, it is surrounded by another layer which is known as root cap. Does not want the root to get, because when root goes down, obviously there are chances of tear. We do not want the root to get damaged. That is why we have root cap as well. Okay, so these children please remember, the dicots that you see, they have two cotyledons, dicot, two cotyledons. Okay, upper side epicotyl, lower side hypo. Hypo means every time low. Hypothyroidism means low thyroid hormone is there. Okay, so hypocotyl is there. Now this whole axis that you see is known as embryonal axis. Towards the lower end you have radicle, towards the upper end you have plumule. Plumule forms the future shoot, radicle forms the future root. Saving the root, you have the root cap. Now on the other hand, when you talk about monocots, they are like, I said no, I do not want to use the complete endosperm. I do not need it. We are not as I don't get the right word, but yeah, we do not want all this thing. We do not want to do all this drama. Instead, we just have one cotyledon, and this cotyledon should be known by all. This cotyledon should be pretty famous because this is the only cotyledon that we have, and that cotyledon is everything has went. Are will it work? Over here, this cotyledon that you see, scutellum, it is known as what? Scutellum. It is the only only cotyledon that it has. Cotyledon that it has, not cotyledon. Cotyledon that it has. And also I said there will be some amount of endosperm left. So this is the endosperm that you see over here, endosperm that is left. Now here also they will have epicotyl and hypocotyl. The upper end will be known as plumule, the lower end will be known as radicle. And surrounding them there is undifferentiated sheath which is known as coleoptile. So the plumule is surrounded by coleoptile. Over here the lower part is surrounded by something that is known as, where is it? Coleorhiza. Okay, it is known as coleorhiza. I'll zoom out. Okay, see. So there is radicle. Beneath radicle you have root cap as well. Beneath radicle you have, children, the root cap as well. Okay, and then beneath it you have coleorhiza, that is undifferentiated sheet. Upper they have coleoptile. Okay, they have coleoptile. This is how monocot embryo is different from dicot embryo. So initial stage of development will be same, but later on it will be different. Now they will ask you, what is the non-albuminous seed? What is the major storage? How do they majorly store things? Non-albuminous, as in they do not have endosperm. So they store in the form of cotyledons. On the other hand, if they ask about them, then what do you say? It will be endosperm, because that is why the endosperm is still there. Okay. Examples, learn muddy. This also learn muddy. Okay. Going ahead, yeah, there is one more type of seed apart from all this thing. There is one more type of very important seed. So you know, remember the nucleus, the cells of the nucleus. Among them, one was forming the megaspore mother cell. The nucleus is still there over here. The nucleus layer is still there over here, and it is doing what? The cells of the nucleus is going to provide nourishment. The cells of the nucleus are saying, we will also provide nourishment. We cannot leave you like that. We made you. We cannot leave you like that. We will also provide nourishment. And now this layer of nucleus which is there, it is known as perisperm, children. What do you call it? Perisperm. And these type of seeds, what do you call it? Perispermic seeds. What do you call it? Perispermic seeds. The examples are beet and black pepper. So just imagine beetroot and black pepper, you're mixing it and eating it, you're not liking it. But still, it was made with love, so you should be liking it, you should be eating it, it's a very good thing. Now over here, the nucleus, the layer which is forming is known as perisperm, and these plants, seeds are known as perispermic seeds. Example is beet and beetroot, please do not forget this, okay. Done with types of seeds, dicot, monocot, and then perispermic seed is also there. Yeah. Now types of fruit. So usually I said ovary, after fertilization the ovary itself forms the fruit, that is the true fruit, that is naturally happens. Now there are another type of fruits also, where the ovary says, I cannot leave thalamus and come. Thalamus and I have been living together since long. So as of now the thalamus also comes in. Or any other fruit part apart from, any other flower part apart from the ovary, if it is also joining together to form the fruit. Okay, it is also joining together to form the fruit, you call them as false fruit. Okay, what do you call them? False fruit. For example, apple, cashew nut, strawberry, they all come in this category. There the thalamus is also included. Thalamus, you remember thalamus on which it was resting in the flower, basically it is also included. So remember, it's along with the ovary, these parts are also contributing to the formation of the fruit, which is why it is known as false fruit. True fruit is only this. False fruit is also this, okay. So we have apple and cashew nut and strawberry. Now that is another type of fruit, okay. There is another type of fruit way beyond our league only. Here fertilization has not only taken place. Now you might be thinking, ma'am, too much of bio, we cannot take it. How without fertilization, how the seed will be formed? You only tell. How, if seed is not formed, how will fruit form? That is another story. If the seed is not formed, how will the fruit form? Usually what happens, the fertilized ovule, which is known as seed, it releases some hormones, which is why the ovary becomes the fruit. Now if it is not being there, then how it happens? But it happens. In biology, everything is possible. So basically it also happens. So without fertilization, the ovary, because of some hormones, of some chemicals being released, okay, naturally also it happens, okay. And since it is occurring without any fertilization, so there won't be any seeds. There won't be any seeds. Example is banana. Example is banana. But banana you make like this with the help of inducing auxin. You put auxin from outside. Okay, you put auxin from outside. Auxin is a hormone, and when you put it, this particular ovary that is there, it slowly turns into a fruit, and which is why there won't be any seed. Just like that, you have tomato as well. You have seedless tomato and things like that formed because of this, and these fruits are known as, children, parthenocarpic fruit. Please remember, fruit without fertilization is known as parthenocarpic fruit. This is where we, parthenogenesis is different, please remember that. Parthenocarpic fruit means fruit without fertilization. Without fertilization. Okay, that is parthenocarpic fruit. Example, banana. Okay. Now children, are you enjoying this class, or is it me just talking? Today I am very energetic because as you know, I made this notes with so much amount of love. I do not usually get this much time, or else I would have made so many notes for you. So yes, today I was finally able to make this, and I think that this will be useful for you when you read it, right? So if you're liking it, do let me know now only. I will be reading down your comments and all. So I'll be so happy. Anyways, next thing. So there is something very different which is known as apomixis, and it is known as, and also polyembryony. This is two different terms. So apomixis is basically, we learned about how we can produce fruit without fertilization. Now they are saying, parthenocarpy, please don't even come to fight with us. We are going to form seed without fertilization. Now that is something unique. If you are forming seed without fertilization, that means embryo should be there inside. That's when the seed will be forming. You call it a seed when embryo is there, right? So apomixis is seen in family of Asteraceae and also grasses. Okay. Basically what happens, first scenario is when there is a female gamete, but this female gamete, you know how it is produced usually. Megaspore, mother cell is there, it undergoes mitotic, initially it undergoes meiosis to form the megaspore, right? There will be a megaspore which is formed as the end result of meiosis. So that means haploid number of chromosomes are there. Now this particular cell undergoes nuclear division and there will be 8 nuclei. Right. So one nuclei among them will be of the female gamete, it will be haploid. But over here, just imagine that the female gamete has been produced but without the reductional division. Without meiosis, the female gamete has been produced and hence this will be diploid. This particular female gamete, it will be diploid since reductional division was not taking place. Now what will happen? This will say, I do not need male gamete, I am already diploid, I can just divide my cells, undergo mitosis to form a diploid embryo. This is one type of apomixis. Okay. Now in the other type, again the nucleus cell which was there, which was forming perispermic, seed and all, if you remember. Here also some amount of nucleus is left, and what is this nucleus is doing? The other one was just providing nutrition. This nucleus is saying, I cannot stay outside, I want to get inside the embryo sac and see what's happening there. So while it gets inside, it will form, it is small cells, no? So for getting inside, it divide. Now you have multiple cells over here, multiple cells over here, multiple cells over here. Okay, now these cells are going to form the embryo children. Don't ask me how, why and why not. It happens like that, and this is known as apomixis. So there will be so many chemicals and hormones being released which would be leading it to an embryo, but yes, it definitely forms an embryo. So over here the cells of the nucleus get inside the embryo sac and forms multiple embryos. Now this case where multiple embryos are formed, you call this polyembryony. You call this polyembryony. So because of apomixis, second type of apomixis, polyembryony is been happening. Examples are mango and the other examples are citrus fruits. So many questions have come. What is happening in citrus fruits? What is the one that forms the polyembryos, so multiple embryos and stuff like that? It is the nuclear cells. It is these cells or the nucleus or the nuclear cells. Okay. Now one more thing, they are also known as sporophytic cells because they are going to form the spores, right? Megaspores later on. They are also known as sporophytic cells. Don't worry, nucleus is also known as sporophytic cells, the same thing. Just understand. And it's all happening in the ovule. The whole thing is happening in the ovule. Please remember the example mango and citrus fruits, they have asked so many times. Okay, now whenever your zygote is not, instead of zygote, something else is forming the embryo. Actually, this is known as adventive embryony. So instead of actual zygote, by the fusion of male and female gamete, you get zygote, right? So instead of zygote, if someone else is coming and form the embryo, you call this adventive embryony. And they might be asking, adventive embryony is seen in this is plant, and one option is mango. Go with mango. That's correct. Okay, citrus fruit, that is also correct. Okay, so now we spoke about seeds as well as fruits as well. Okay, am I forgetting something? Did I miss something up? Yes, I missed the structure of the seed. Let me talk about the structure of the seed also. Where can I talk about the structure of the seed? Why is it not going now? Okay. Okay, here I will talk about the structure of the seed. Okay. So this is a seed which has the embryo being formed over there. You saw how the embryo, I am making a dicotyledon embryo. So this is a dicot embryo that is being formed over there. So the seed basically has seed coat, children. So the inner integument which was there, remember, in the ovule, it is going to form a layer which is known as tegmen, and there will be an outer layer which is formed at the outer integument. You call this testa. What do you call this, children? You call this testa. Okay, so you have tegmen and you have testa. This is the inner layer, this is the outer layer. Okay, happy? And there will be a pore also which is in the micropyle region only. This pore will provide all the nutrition and things like that. Air, water, things like that. Okay. Now one more thing, dormancy. The seeds can be dormant. When you say it's dormant, basically when the metabolic activities, it's very low. What did I say now? What is the term? Seeds can show dormancy. Basically when the moisture content below in them reduced to 10 to 15%. It reduces 10 to 15% reduces, and the metabolic activity is very less. Metabolic activity becomes very less. Okay. Now over here, where am I? Array, it's moving sometimes. Okay, now we are done. Now we have to do seed viability. Now these are the examples that is given in NCERT, so we have to read it. Just before that, let me see what's the problem over here. Yeah, so basically seed viability, there are two plants examples. Lupine, that is given over here in the NCERT. It said that for 10,000 years the seed remained viable. What do you mean by viable? That basically if you put the seed now in the soil, it will germinate. That is what you mean by viability. It has the power to germinate, to give rise to a future shoot and the root. Okay, so lupine, basically it was viable for 10,000 years, and the plant's name is Lupinus articus. The plant's actual name is what, children? Lupinus articus. Now there is another plant, that is Phoenix dactylifera. Okay, it was also viable for 20,000, it's 2,000 years. Okay, it is known as date palm, children. So you have Phoenix dactylifera, which is date palm, viable for 2,000 years. Lupine, which was viable for 10,000 years. This is Lupinus articus. It is known as Lupinus articus. Seed viability is very much important. Please remember, all here in plants, basically you have to remember the examples. That is the most important thing. The same thing being repeated over here. Now let's talk about, we are done with seed, we are done with fruit, we are done with everything, all the female and the male parts. Now the last part of the chapter, children, I have made it last. Okay, it is known as pollination. It is known as pollination. What do you mean by pollination? Transfer of pollen from the anther to the stigma, right? Simple. But let's make it complicated because that's biology all about. So autogamy is there. There are different type of pollination. One is autogamy. As you can see over here, when pollination is happening in the same flower, children, when it is happening in the same flower, it is known as autogamy, and is also known as self-pollination. Self-pollination. But there are two conditions for self-pollination to happen. First thing, the pollen release and the stigma receptivity should be synchronized. As in, when the pollen is releasing, the stigma should be actually receptive. If it's not receptive, will it land and form and germinate and do all this thing? No. So that is one thing. Okay. Second thing, position of the anther and the stigma. Someone is here, for example, the catcher is somewhere else, and the thrower is somewhere else. The bowler is somewhere else. I'm putting my ball over here, the catcher is that side. Will it work? No. So position of anther and the stigma should be nearer such that pollination could happen. So these are the two conditions for autogamy to happen. Okay. Now autogamy is shown by these type of plants, okay, which has exposed anthers which are bisexual in nature. The flowers are what, children? Bisexual in nature. As in, they should be having two sexes together, the male and the female reproductive part. Okay. Now in your NCERT there is an example given. Let me just a second, children. I will die of cold. The AC works now. Yes. So in your NCERT there is a plant picture that is given. I don't know if plant picture is given, example is given. There is Viola, Oxalis, Commelina, all different names of something big. Viola, Oxalis, Commelina, they have two type of flowers, children. One flowers are like this, okay. One flowers are like this, where the anther and the stigma, everything is exposed. Anyone can do pollination over here. They do autogamy, but other pollens can also, obviously if there is some other plant, it will also produce its pollen grain, and this pollen grain can also land over here on the stigma. So they have exposed anther and the stigma, and these type of flowers are known as chasmogamous flower. What do you call it? Chasmogamous flower. But there is a twist. In the plant body, in the lower region, they have closed flowers where the same thing is there and they are closed. Now you think they are closed and you see anther and stigma. Obviously, if you have everything over here, you will do fertilization here itself. Can you get pollen from outside? No, because it is closed. There is no option for cross-pollination. Okay, over here, self-pollination is pakka, sure thing. And one more thing is sure. Fertilization will happen, I will get my seed. So I say in these type of flowers which are known as clistogamous, clisto, okay, CL for clisto, CL for close, I always say it. So clistogamous flowers are the one which will give me assured seed set anyways, the seed set will be produced. And second thing will be, it will be invariably autogamous. What do you mean by invariably autogamous? That means No choice given. It has to be autogamous. Autogamous as in it will be showing self-pollination. But over here, even though it is an autogamous plant, over here it can show autogamicity, but still other flowers can also give their pollen grains and still things can happen. So one is chasmogamous and the other one is clistogamous, a short seed set, and invariably autogamous. Example, Viola, Oxalis, Commelina. Viola is also known as common pansy. Okay. Next thing, guide-to-nogamy, where there is the same plant, if you see, but different flowers, not the same flowers. If the same flower, it is self-pollination. Over here, they are functionally cross-pollination, exactly, cross-pollination is happening functionally, genetically autogamous, because the same plant, same genetics, nothing different. So genetically, it's again a very important point. Genetically, they are autogamous, but functionally children, they are showing cross-pollination. Okay, now cross-pollination between different flowers of the same plant, that is guide-to-nogamy. Third thing, still not working. Okay, it's not also not working. Oh, sorry. Yeah, xenogamy. What do you mean by xenogamy? There are two different plants, as you can see, two different flowers, okay, and they are doing pollination. So pollination between different flowers of different plant. Obviously now here genetics would be different. So you have one male person, female person, two from different parents. Obviously the progeny will be having different genetic variations. Similarly over here also, it's pakka cross pollination and it will be genetically different as well. That is xenogamy. Okay, agreed? Now going ahead, you have two plants. Okay, one type of plants are known as monoecious plants, another one are known as dioecious plants. Monoecious are the ones where you will find both female and male flowers. Okay, you will find both female and male flowers. On the other hand, dioecious is when you have a plant, you have another plant, and now over here you have flower. Okay, and there will be one female flower, one female plant, and the other one will be what, children? Male plant. So can I write separate sexes in separate flowers? Is it okay? Separate sexes and separate flowers. Over here you have both the female and male flowers in the same plant. Okay, this is kind of different, this is also kind of different. Now there is one more, so we are talking about unisexual flowers. So one flower, if you see, it will have only one sex, but if you see as a whole, it will have two sexes. Okay, so basically this flower is having female flowers, the other one is having male flowers. Okay, whereas over here it is the female plant and the other one is the male plant. God. Female plant and this one is the male plant. Okay, now examples: coconut, castor, cucurbits, 3 with C's and 1 M. Okay, CM, you can remember like that, chief minister. So coconut, castor, cucurbits and maize, and over here you have papaya and depa. Now will the monoecious flower, which are unisexual in nature, I am not talking about bisexual, there would be bisexual flowers as well, where there is anther and stigma, both of them together in a flower. I am talking about unisexual flowers. Okay, over here, do you think autogamy can take place? Do you think autogamy can take place? No, children. For autogamy, you need male and the female in the same flower, but they are unisexual flowers. So autogamy cannot take place in monoecious unisexual flowers. Okay. What about guide-to-nogamy? Can it take place over here? Guide-to-nogamy is basically when male to female, or basically when between flowers of the same plant it is happening, it is known as guide-to-nogamy. So guide-to-nogamy, it takes place over here, so which is why I made it tick. Xenogamy, what about that? So from this plant, okay, so from this plant, if things are going to another plants, unisexual flowers, okay. So then also xenogamy can take place. Another plant is there, so xenogamy can also take place. But what about them? What about them over here? So Autogamy, do you think it can take place? Again, it's a very big no. Autogamy cannot take place. What about gytonogamy? From here to here, if things go, will anything work? No, children. Both of them have the same sex. So gytonogamy is not possible. Xenogamy, if you talk about that, xenogamy, is it possible? Yes. From here to here, things can come. So in between plants, this is happening. So xenogamy is the only possible thing in dioecious plants, children. Xenogamy is the only possible thing in dioecious plants, and papaya and date palm is a very important example, especially papaya. So many questions have asked. Okay, especially papaya, so many questions they have asked. In papaya, xenogamy is possible, gytonogamy is not possible. Xenogamy is possible, autogamy is not possible. Options would like that would come. You have seen the options. I have made you solve the questions as well. Now you will be seeing in mock test as well. It is tomorrow. So basically, in dioecious unisexual flowers, things like this happen. In monoecious unisexual flowers, obviously autogamy does not take place. Both of them, they take place. Works for you? Examples, very, very important. Okay. Now, going ahead. Okay, now there are different types of agents for pollination. Okay, there is biotic agent, there is abiotic agent. Biotic agent as in insect, animals and stuff like that. Abiotic as in wind, water and things like that. So, majority of the pollination happens because of the biotic agents, that is insects. Okay, but if you talk about abiotic agents, in them wind is better than water. Okay, wind pollination is better than water. Now these lines are exactly from your NCERT. I just did little bit cut up it, that's all, because all the points of your NCERT are really, really important. So for wind pollination, what are the criterias? Pollen grains has to be light and non-sticky. If it is sticky, then obviously it will go stick there, it won't move around. We do not want that. In the wind, it should move. So it should be non-sticky and should be light. If it's high weighted, then obviously it cannot move around. Okay, that is one thing. Well exposed stamens, children. The stamen should be well exposed such that when wind comes, all the pollen, it goes along with it. Okay, so stamens should be well exposed. Next thing, large and often feathery stigma. So the stigma is not feathery, things won't come and attach to it. The pollen grains are flying. If they go flying like that, there is no possibility of pollination happening. We want it to come and attach. So basically for that, we need feathery stigma. Large and often feathery stigma we need. Next thing, wind-pollinated flowers only have a single ovule. So if you talk about a flower, single flower, they will have a single ovule. Why? Because they cannot guarantee that the pollen will come and land over it. If I make multiple ovules and I sit, I will be in depression. I do not want that. I'll be really sad. I do not want that. Okay, so depression is a very big word to use. Sorry for using that. So basically, I'll be really, really sad. I do not want that. So basically, wind pollinated flowers cannot lose their energy in making multiple ovules. Instead, they make one single ovule. Now, if there are flowers, okay, they make a bunch of them together. You call this as inflorescence. Okay, the flowers, they come in bunch, you call them inflorescence. Why do you think it's happened? Because if one flower is not able to catch the pollen, the other flower can pollinate and then the whole process can happen, which is why inflorescence is very much unique in them. So inflorescence, having a single ovule, all these unique features you need to remember. They'll ask you for a wind pollinated plants, what are the features? So you have to tell. Large and feathery stigma also. Okay, now wind pollinated is quite common in grasses. Please remember grasses. Wind pollination is very common in grasses, and also the corn cob that you see, okay, the tassels that you see, children, it is nothing but the stigma and the style. Please remember this also, stigma and the style. Okay. And wind pollination is known as anemophily. Do not remember animal with this. This is anemophily, something different. That is when animals are doing something, as in big animals, I am not talking about insects. It is known as zoophily. Okay. Here it is known as anemophily. Don't worry. Now, water pollination as a whole, it is very much important. So, they are saying in water pollination, I have read it out when we were talking about that. But still, about 30% genera, okay, so 30 genera, mostly monocotyledons do this. Okay, 30 genera and monocots, they basically do this. Now, for some others, for example, lower plant groups such as algae, bryophytes and pteridophytes, which I have written over here, for them this is a regular thing. They actually live in water because they actually do not live because if water is not there. Such importance is given to water. So basically 30 genera of monocotyledons, they do it. And one more very important thing is that most of them, they are children monocots. Most of them, they are monocots. Going ahead, only lower plant groups can do it, such as algae, bryophytes and pteridophytes. Okay, now going ahead. There are certain plants, for example, water hyacinth and water lily. Okay, water hyacinth and water lily I want to make over here. So what about them? What's very unique about them is that there is water, and these guys, they come over here, they have the flowers at the top, okay, they have their anther and everything there visible, but they do not do water pollination. Instead, they wait for wind or they wait for insects for pollination to occur. So please remember, water hyacinth, water lily, both of them are in water, but they are relying on insect and wind for pollination. Talking about Vallisneria, okay, Vallisneria is basically another plant which lives in water, and I'll tell you their story. Okay, they do water pollination, don't worry. So they come up like this, and only the one that has the flower reaches like this, and now it has the flower over here, okay, with the female reproductive system. Now there will be another plant over here, the one with the male reproductive system, okay. Now either the pollens are released over here in water, and the pollens, they swim and come, okay, or the flower itself is dropped in water such that the flower comes in contact and they do pollination, in the water itself. Basically the pollination happens, and this is happening because of water. If water is not there, the pollination is not usually occurring. Okay, I'm telling you again, over here the pollen grains are either released over here, okay, and do not expect them to be in this much height. You can think like that. Okay, you can think like that, it's not in this much height. So basically the pollen grains are released, or the flower itself is released and comes there, and then they pollinate, and then later they come inside and then they do fertilization, whatever process is there. It can be taken inside as well. That is about Vallisneria. Now talking about sea grasses, they are very shy. They are very shy. They say that we would be just doing pollination within the water itself. So there are sea grasses, okay, you call, example is given in your book, Zoostera, okay, they have their flowers over here and they pollinate inside the water only. But here one very important thing is that their pollen grain will be ribbon-like. Will be like ribbon-like. What, children? Ribbon-like. Okay. Along with Vallisneria, there is one more example. Wait. Yeah, Hydrilla. Hydrilla and Vallisneria is there, both of them. They reach the surface of the flower by the long stalk, and the male flowers of the pollen grains are released on the surface of the water. So over here they reach, and then they release the pollen grains in the water and also flower in the water. Okay, this is about Vallisneria and Zoostera. Rest is about Vallisneria and Hydrilla. Vallisneria and Hydrilla. Rest is about Zoostera. It has submerged. It does submerged pollination. Submerged pollination. Okay, when they are submerged pollination, obviously these pollen grains have to move, which is why they are ribbon-like. Now one more thing, when pollen grains are in water, they are covered by a mucilaginous covering which does not allow their decay, otherwise they will be getting decayed. Okay, now the last thing, children. The last thing, insect pollinated flowers. Okay, talking about them, let's talk. So if you are an insect bee, okay, then for you the plant should be looking really pretty, which is where you go. So this, and it should be visible more than being pretty. Okay, so basically this insect pollinated flowers, they are large, they're colorful, they're fragrant, such that all the insect can come there, and it will be rich in nectar as well, because the insect is not cupping for its beauty. Insect wants the nectar. Okay, insect wants the nectar for its own purpose. Now when the flowers are small and the number of flowers are clustered into inflorescence to make them conspicuous. Basically, if flowers are small, what do? Again, you make them into cluster and form the inflorescence such that it will be visible to them as well. Now this is a feature that you saw in wind pollination, but in wind pollination things were different. They were not rich in nectar and things like that. Over here, they are rich in nectar and they are brightly colored and stuff like that. Please understand the difference. Now the flowers pollinated by flies and all, they do not need fragrance as in good fragrance. They need bad foul smell, so that things can come. You know Rafflesia, I've spoke about Rafflesia. This has a very bad smell, like a rotten egg. and hence the flies and everything comes and pollination happens. So huge flower. Okay, now going ahead, nectar and pollen grains are the usual floral rewards, and there are certain things, certain insects, they are so shy, they come in, okay, they come in the flower and they take the pollen and they run away. They are robbers. They are robbers, okay, the one that robs the pollen and the nectar. Now in some species, floral rewards are in providing safe space to lay eggs. An example is that is the tallest flower, Amorphophallus. This is the Amorphophallus flower. What is happening? Instead of giving nectar and all, they say, you come here. You lay your egg only here. If you lay your egg here, you can grow. I'm providing you a safe space, but please take my pollen grains. So what happens? That insect comes in, sits on it, sits on any flower, and then pollen grains, obviously they are sticky. These ones, okay, they will be having sticky pollen grains. Please remember. Sticky pollen grains. So pollen grains will now stick on it. Now it will move around, go to some other flower, give that pollen grain there when it moves there. Okay, so usually nectar and everything is given, but over here they are giving a space to lay eggs also. This is in the case of Amorphophallus, which is the tallest flower. I was talking about largest flower, that is Rafflesia. Okay, now over here there is one more relationship that exists between both moth and plant yucca. This is moth and this is the plant, that is yucca. Both of them, they are in an inseparable relationship. Okay, they are together always. So moth basically goes and lays its egg where? In the locule of the ovary. What is locule of the ovary? By that I remember, the space inside the ovary where the ovule is resting, you call it a locule. So inside the locule of the ovary, this moth goes and lays eggs, and later when the seed starts developing, okay, later when the plant, this pollination and everything happens and the seeds start developing, that's when these eggs are coming out and larvae is going to come out basically, not egg cannot come out, larvae comes out from the egg. That's a very, very, very unusual thing, right? So that is between the plant and the moth yucca. Now children, when insects are doing pollination, you call it entomophily. When animals are doing pollination, you call it zoophily. Okay, all these terms are really, really important for you. Now one more thing, it's not just insect, and by animals what was I saying? I was meaning lemurs. Okay, sometimes reptiles also, like lizards, and this question has come, is reptile a pollinator? Yes, sometimes reptiles also act as pollinators. Okay, the birds, everything can come under this category. So with this, we have completed the sexual reproduction in flowering plants, my dearest children. I hope that this particular session was really, really useful for you. We spoke a lot, but by speaking a lot, we have covered a lot as well. Now, do you think that you are ready for tomorrow's mock test? I believe that you are perfectly ready, but yes, you need to revise a bit. Go read your NCERT, solve certain questions. I would say you have already read your NCERT with me. I have already covered a lot of things. Please solve certain questions. That will be more helpful for you. And then if you think you are not confident, then come to NCERT. But solve questions. Go to Google and type sexual reproduction in flowering plants question and try to solve few questions and see if you are able to answer them or not. Okay, so that's all, children. Do you think that this particular video was helpful? Do you think that we are helping you enough? If you think that we are helping you enough, do not forget to subscribe to our channel, that is Diksha Karnataka, and do not forget to leave the comments down. And I wish you all the best for tomorrow's mock test. Whatever comes your way, even if you do not score really well, it's fine. You try your level best. That's more than enough. Okay, and let's see who all can get the good marks. Okay, but I will be happy even if you are just trying, and even if you are not getting good marks, is fine. Do let me know. In whatever ways I can help you, I will definitely help you out. Okay, so bye bye children, have your sweet little smile and have a really, really good day. Take care.